Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you another really fascinating guest today involved in creating a better tomorrow for all. Today, we have the honor of being joined by Dr. Khalid Salaita, who is a professor of chemistry at Emory University in Atlanta, program faculty in the Department of Biomedical Engineering at Georgia Tech and Emory, program manager of cancer cell biology at the Winship Cancer Institute, and most recently, the winner of the Future Insight Prize given by Merck KGaA at Darmstadt, Germany, for his cutting work in the area of mechanobiology. Dr. Salaita earned his bachelor's in chemistry from Old Dominion University, his Ph.D. in chemistry from Northwestern, completed his postdoc fellowship in the Department of Chemistry at University of California, Berkeley, and then started his own lab at Emory, ultimately to investigate this really fascinating interface between living systems and biology and what they call engineered nanoscale materials. And to achieve this goal, his group has pioneered the development of numerous tools like molecular force sensors, smart therapeutics, nanoscale mechanical actuators to help manipulate living cells, and ultimately the term DNA Mechano Technology, which we're getting into. And in recognition of his work, Dr. Salaita has received numerous awards, most notably the Alfred P. Sloan Research Fellowship, the Camille-Dreyfus Teacher Scholar Award, the National Science Foundation Early Career Award, and the Kavli Fellowship. Dr. Salaita is a current member of the Enabling Bioanalytical and Imaging Technology Study Section, and associate editor of Smart Materials, and his programs have been supported by a range of organizations, including the National Science Foundation, the NIH, and DARPA. And then just a quick note, the Future Insight Prize was set up by Merck KGaA Darmstadt, Germany, to ultimately stimulate innovative solutions to solve some of humanity's greatest problems and to realize the dreams for, as we say, a better tomorrow, and as they say, a better tomorrow in the areas of health, nutrition, and energy. A lot of really exciting topics to get into today. We're honored to have him. Dr. Khalid Salaita, thank you so much for taking the time to come on our show today.
Thank you so much. I'm really looking forward to this. I appreciate that introduction. Really nice.
I have been looking forward to this one as well, because the intersection of biology and physics is something really near and dear to me as sort of an evolving space in the life sciences. I'd like to take us back a little bit, because going through some of your sort of early materials, I took a look at your PhD, this dip pen nanolithography, from fundamental transport processes to high throughput applications. You worked on plasmonic nanoparticles during your undergrad. You did postdoc work in receptor clustering and cell signaling. A lot of really interesting topics. Talk about how this all got started, because it is a really unique area, to say the least. And I'd love to hear sort of about the early days and what got you interested in this whole domain.
Oh, wow. You're taking me back a good 20 years back in history. So I would say it all started when I was an undergraduate. And at that time, I became aware of this really amazing area of nanoscience. And there was one fact that just blew my mind. And that is, if you take a piece of gold and you measure its melting temperature, it has a melting temperature that's close to about 1,000 degrees Celsius. But then as you shrink it down into smaller and smaller bits of gold, you get down to the nanometer length scale, that melting temperature suddenly changes. Gold that is nice and yellow suddenly becomes a red colloidal suspension when you get down to gold that is small enough in dimension. And to me, as an undergraduate, that just shocked me. I couldn't fathom how that's possible and why the rules were changing on us. And that got me started doing the undergraduate research, really studying the properties of small plasmonic particles like gold particles and silver particles. And during that time, I read an article in Science from Chad Merkin, who later became my PhD advisor at Northwestern. And he had written an article describing the world's smallest pen and using the world's smallest pen to deposit and write nanoscale features on a surface. That got me even more excited about the ability to manipulate matter at that length scale. And so that led me to doing a PhD with Chad, which was a lot of fun. And that article was as advertised. I really got to use this machine that controlled a small cantilever, which provided a really sharp point that you would load with molecules and deposit them and ask really basic questions. How does matter change? How do the properties, the electrochemical properties or other properties of material, how do they change when you change the way that they're organized at these smallest length scales? At the end of the PhD, what led me to doing a postdoc with Jay Groves at UC Berkeley, well, it was happenstance. A lot of what, the progression through science, it's out of curiosity and happenstance. And Jay was giving a talk, and he was describing how the surface of a cell has very specific arrangements of receptors. And he was talking about immune cells and how the arrangements of their receptors at the nanometer length scale completely changed decision-making. And again, that triggered all sorts of questions in my mind. How does a cell distinguish between these molecules that are studying its surface, how they're arranged in a particular pattern? How does that change outcome and behavior? And I thought, given that I've been studying how to organize materials at these small length scales, I thought it would be a natural transition to study living systems with molecules that are arranged in certain nanoscale patterns, and that led me to my postdoc, and the rest is history. And I started at Emory several years after that.
Awesome. And continuing along that timeline and that school of thought, so you spend your time learning and thinking about sort of these nano machines, these nano devices at the nanoscale. You enter the world of sort of the life sciences, and we are at this fascinating period right now, as you were saying, whether we're talking about that drug hitting the receptor or that cancer cell taking it's sort of the beginning of its metastatic journey. The interest in some of these physical themes, whether they're membrane potentials, forces of mechanotransduction, and so forth. There's a lot more going on at the level of the physics, and sort of this, all falls within everything that you ultimately are developing. Talk a little just about sort of the era we're in, and then we'll get into some of what you've been doing the last couple of years. But in general, sort of the general feel of the area nowadays, as physicists are beginning, or biophysicists, let's say, are beginning to talk to the molecular biologists, to the pharmacologists, and so forth. What's the sort of the scene like nowadays with regard to the merging of these tools?
Yeah, no, that's a really good question. I think for a very long time, the way we understood living systems and biology was from the lens of taking cells or tissues, putting them in a blender so that you mix everything up, release all the biomolecules in there, and then we conduct experiments to understand how the different components, the different enzymes and nucleic acids and lipid components, how they function. And so for the longest time, we viewed the cell from the lens of essentially a sack of molecules that are homogeneously distributed and having these functions. But what's increasingly becoming clear is that it's not a random arrangement of molecules. That the biomolecules inside a cell have very well prescribed zip codes, very well orchestrated arrangements of whether the molecule resides at the cell membrane or associated with certain organelles or associated with certain neighborhoods of other proteins and biomolecules. And so suddenly these tools that were developed originally in the material sciences, for example, or by biophysicists, tools to identify the location and the dynamics and where molecules move from point A to point B, where they reside inside the cell, that is increasingly becoming a much more important problem. And then the additional layer of really interest that's rising is not just the location, the spatial arrangement of where these proteins are, but also their mechanical state.
Yep.
So if you let a biomolecule loose inside a cell, it's constantly getting bombarded and bumped around by water molecules or other proteins or other biomolecules, and so it randomly diffuses, it randomly hops around like a drunk sailor. But it turns out that that's not the case for most biomolecules. They either are attached to a membrane, so something like half the known proteins are predicted to be associated with a membrane. So they have a membrane interacting component at some point in their life cycle. They're attached to a membrane, and so that on its own means these molecules aren't diffusing in 3 dimensions. They're skating in 2 dimensions on a sheet of membrane. And then moreover, these biomolecules many times are being shipped and moved around intentionally with things like motor proteins that will use ATP and shuttle either the protein itself or a vesicle from certain locations and other locations in the cell. This is maybe not a surprise to us. It's intuitive. It makes sense. I mean, if you look at the shape of any organism, it has a characteristic shape from a snail to a fish. And even if you go down to the scale of the tissue itself, each tissue has a characteristic architecture. But even if you drill down to a single cell, you look at it under a microscope, it has very unique shapes. And in fact, that's what a pathologist is trained to do, is to look at the shape and the organization of a cell and use that information as a predictor of a disease state.
Yep.
So to some degree, it should be obvious that stuff is important. But only recently, I want to say, recently in the last few decades, have the tools come around so that we can start interrogating, let's say, what level of mechanical forces, where is the exact spatial coordinates of a protein within a cell, and how these spatial coordinates impact function. These are quickly changing our ability, and so I think it's a long recognized black box that's very quickly getting revealed with the advent of these physical tools.
And in lieu of all that, you have spent the last couple of years now in creating, what we'll call these DNA based machines that, can do everything from walk to compute to sense to deliver stuff. I'd love to take a walk through some of your publications on these topics. They're so, each one is so elegant in their own, but sort of how these have evolved. And I think an important one to start off with would be your Nature Nanotech paper from 2016. This is a high-speed DNA-based rolling motors powered by RNase H, where you first sort of describe this I sort of visualize it in sort of the little video, so there's sort of like a Pac-Man type DNA motor sort of rolling along, moving around. Talk about this particular piece of research. I think in the scheme of moving and then delivering stuff and sensing stuff, it's the beginning of a very interesting story.
Absolutely. So let me start, I guess, with what led us to that paper in the first place. I started off by talking about the fact that biomolecules in the cell are not randomly organized, and they get moved around or shipped around, and the way that happens is through biological machines and biological motors that use ATP and convert that into mechanical work through a stroke mechanism. So each ATP molecule that gets consumed triggers a conformational change, a wiggle in the arrangement of a motor protein. And if that wiggle happens on a track, that leads to locomotion. And so for a long time, the physical scientists, chemists, and physicists, we've always wanted to create machines like the biological machines, like the ones that were created by nature, with the idea that if that's a really basic property of living systems, and if we can start to create synthetic systems that can do that can take in chemical fuel and convert that into mechanical work, that takes us a step closer to understanding life and maybe creating new technologies. And so some of the earliest synthetic machines created were actually made out of DNA. And while the natural machines are not made out of DNA, natural machines are made out of proteins, and they're all enzymes, in fact. But the reason that we as in the field started off with nucleic acids with DNA is basically because of the well-known rules of how to assemble DNA together, and how to get something like a piece of DNA to have a repetitive cyclic type conformational change that could lead it to this progression toward locomotion, toward a motor. And so what the literature had, we had saw in the literature are sets of DNA machines that were typically bipedal, so meaning two legs. Okay, and the bipedal motors, they're typically often they're described as having two legs, and each leg would have a foothold on a track. And by adding different components in solution, you could trigger one leg to move, the other to stay still, and then to flip that so the back leg is released such that it moves forward, and what was previously a forward leg becomes a backward leg. You can then go through a cycle of doing a bipedal, just like you imagine a human being taking walks. You do this bipedal motion. And so those are the initial kind of where the field started with these DNA motors, and this is where we started looking and thinking, could we make a more efficient motor? But the reason we were asking whether we can make a more efficient motor is because those early motors moved at speeds like a nanometer per hour. Okay, so you would spend overnight to observe one of these DNA walkers moving something on the order of 10 to 20 nanometers. And again, often you'd have to use an electron microscope or another really high resolution type microscope, like an atomic force microscope, for example, to even see the motion. And so we looked at it, and we saw that there was really a fundamental trade-off to create better motors. And the trade-off was that if you wanted to create a faster motor, all you had to do is to speed up the step process, each stroke. But the problem is, if you sped up the stroke process too much, there's a probability that both legs let off the track. And if you're a macroscopic object, you and me, both legs are off the ground, you go back to the ground, no problem. But if you're an object that's like a piece of DNA at really small length scales, once both legs are off the track, you are lost to Brownian energy. You're swept away by thermal energy, and that molecule will never come back. That motor will never come back. It's swept away. And so that reduces processivity, the ability to repeatedly continue the motion, or the persistence of a machine. So it was this trade-off. So if you wanted to have more speed, you're going to lose persistence. But you can, of course, have more persistence because you can design a motor with more legs. And so, in fact, somebody had published a spider, which had four legs. And the spider, which I guess doesn't make sense for why a spider would have four legs, but that's what is described in some of those early papers, is that it definitely did have better persistence, but the problem is with more legs, you go slower. And the analogy is like when you have a potato sack race. If you can have two legs by two people, but if you don't coordinate well, that slows you down. And the same thing happened when you have more legs on a motor. You can have all the legs you want, but if the legs aren't coordinated together, the ability to move forward becomes equally likely as moving backward, and the speed takes in a bigger hit. Okay, so this is where we landed and where we started adding to this trade-off, this dilemma. Mm-hmm. And so what we did, we essentially, and this is by accident, I'll have to say, later on, we deduced why it worked so well, but initially this was an accidental discovery. What we did is we took rigid nanoparticles, and we later called these the chassis, and this rigid spherical particle, and we loaded the outside of this rigid spherical particle with a lot of DNA, with a really high density of DNA, okay? And then we took a track, or the surface that the motor is going to move on, and we used complementary RNA. And so what happens is when this multi-armed DNA particle lands on the track, you get this multivalent binding, this Velcro effect. All the DNA binds the RNA, and it's stuck. It's not moving anywhere. But then, as you said in your introduction, it's like a Pac-Man action. What we do is we add an enzyme that destroys any of the RNA that's on the chip, on the surface. And so what that means is anytime this motor makes contact, anytime DNA hybridizes to its complement, that complement gets destroyed and consumed. And so what that means is now your DNA is vacant and allowed to seek new, fresh complements, new, fresh Watson-Crick Franklin base pairing interactions, and that motor starts propelling and moving. Now, in our case, we have hundreds and thousands of legs, so our multivalency went through the roof. So based on the old rules, we should have a much slower motor, okay? But we also use an ultra-fast enzyme, this RNase H, that destroys the RNA, and that's orders of magnitude also faster than stepping motion of the bipedal walkers or the spiders in the past. And so the combination of having a rigid body that curves, it means that the legs on the front of the body are coordinated together. We get coordination that way. Yep. We get very fast turnover from the enzyme, and we get this really high persistence because of this Velcro multivalent effect. These three factors, and it's like magic, because when we've gone in and changed the density of the legs or the speed of the enzyme, the motors don't work as well. And it worked really well the very first time we tried it. And again, absolute luck that it worked out the way it did. And that produced motors that I was shocked, because when these motors started moving, the microscope room is right behind my office. The grad student working on this, Kevin, knocked on my office. He's like, "You've got to take a look at this." I walk in there, we take a look in the eyepiece, and you could literally see these motors by optical microscopy. They're moved such large distances, you can see the motion by optical microscopy. And not overnight. This is like over the course of a 10-minute video. We were blown away. I mean, we didn't understand what was going on, and it took another year to figure out exactly why it worked and what was going on. But it turned out to be a rolling machine, a rolling motor. We thought, in fact, we called it the lawnmower project. We thought it was going to look like a lawnmower, like a Pac-Man lawnmower that slides down a carpet of fuel. And the weird thing is that we would see these trajectories in some of the motors that were ballistic. The motors would just travel in straight lines. And the reason we figured out that they travel in straight lines is because we'd have two motors glued together. So there's impurities in the motors. where you have two rolling wheels, but now attached, physically attached. And that would be the analogy as if you had two bowling balls glued together, rolling down a slope. And the only way they're going to travel, the only way if you glue two bowling balls together and put them on a slope, that pair of bowling balls is going to go in a straight line, just because you have two wheels on linked together in a single chassis. And so you get this linear type motion. And we again, using a whole series of other experiments, we ended up confirming that was the mechanism of motion, that it was a rolling type motion, and that was key in getting that translocation. Now here, this is true story. So we showed this to the physicists, and they were blown away because nobody had shown this type of motion before. In particular, there's different kinds of diffusion. So if you take a random diffusion, and I call this the drunk sailor walk. Okay. The drunk sailor walk means you're equally likely to go in any orientation in any direction. Okay, and that's if you put a piece of pollen on water, that would be Brownian motion. Okay. There's also ballistic motion, which I described, which is just going in a straight line. Right. It's very, very rare to observe for a molecule, but nature's produced some ballistic motors, and those are the motor proteins that travel along cytoskeleton highways in your cells. But the type of motion we observed with the single wheels, that was called self-avoiding motion, which means it was between ballistic and completely Brownian. It traveled in any direction, but not backward, because backward is where the Pac-Man enzymes had consumed the fuel. And so you'd go in this self-avoiding motion. Often polymers are described as adopting self-avoiding paths in a solution. They can go in random orientations, but not overlapping with their past tracks, for example. So the physicists were really excited by this. We broke all the Guinness World Book of Records for speed and processivity, and we send this out to Nature to get it published. And the editor for Nature says, beautiful work, but from all these fundamental discoveries, how is this useful? What new capabilities do you now have? And this gives you the state of how high the bar is these days. Right. So we went back in the lab. We said, okay, we can see these motors moving along a track, but what's interesting is we can observe the motion using really simple low magnification optics, and that means anything that slows down the motion could be recorded using a simple optical device, and that's a way of doing detection. And so what we quickly did is we introduced a point mutation in the DNA sequence. And we said, look, a single nucleotide polymorphism, a SNP, is one type of genetic mutation where it's a single mutation, and it's really hard to detect by using hybridization-based assays. It's just not a trivial thing to detect, and it's actually biomedically relevant because many diseases, not a subset of diseases, have point genetic mutation cause, and they could be ascribed to that point mutation. And so we demonstrated that our motor speed is drastically altered if there's a point mutation in the DNA sequence. And we said, okay, this is a way to detect a SNP that would be really hard to detect using fluorescence-based assays. And moreover, we said, look, this is Kevin, the same student that knocked on my door. He ran across the street to a pharmacy, to CVS, where he bought a cat toy. And the cat toy was a laser pointer that your cat would chase around. And he opened up that laser pointer and took off the plastic lens that's used to focus the laser. And so what he did next is he took this plastic lens and he used double stick tape and taped it to his smartphone. And using a lab jack in the lab, we were able to visualize the motors using this plastic lens attached to his smartphone. Of course, in the paper, we did not talk about the source of the lens, but it was a $2 cat toy. That's where it came from. And the really the part that I was most impressed in is this ingenuity of the student and in being able to show that you can do SNP detection using this bright field readout, by using your smartphone camera, recording a 15-minute video, and confirming that the speed of these motors differs based on what the DNA sequence is coding the machine.
Right.
And so that was where we started this story of starting to build these machines. And again, as you said, that was in 2016.
And continuing on that road, it's 2019 where you publish sort of this perspective article in Science, Emerging Uses of DNA Mechanical Devices. You introduce the word of sort of DNA Mechano-Technology to the reader here, and you sort of take us through, all right, here's machines, and then there's sort of miniature machines, cell phones, microphones, and accelerometers on airplanes, all this stuff. Then you take us into the world of the nano machines, and where we have synthetic DNA now, and the ability to build these other interesting microstructures and so forth. There's a lot of things we could potentially make at this level that, again, can sense or generate forces or transmit information and do a lot of other cool things that we might not have thought of yet. Take us into sort of 2019, and then if you could also, because I know there's a paper a little later than this where you start talking about the principle of roller pin motors based on origami, and that's as far as I'm going to go with that. But introduce us to that particular Mechano-Tech device, if you would, because I think that's also really
cool. No, they're completely related, very connected. Yeah. Both that the roller pin device and this area that we're calling DNA Mechano-Technology. Yeah. And so along the way, over the past 2 decades, this field of DNA nanotech has emerged, and DNA nanotech is like this amazing enabling technique to sit down with pen and paper, and in some cases just a computer, design DNA sequences, throw them in a pot, and then have them self-assemble spontaneously into virtually any shape or size, any structure you want, where your material is organized from a tenth of a nanometer from the scale of the nucleic acids themselves, all the way up to the micrometer length scale. Okay, and this is like saying here's a box of Legos, and you can you as a scientist play and have fun and build any structure you'd like. And the late Ned Seaman, who passed away recently, really is the grandfather or the pioneer of this field, and this has really given everybody the ability to, from scratch, pen and paper, design any structure they want. And so we saw this emerging field, and we saw that we've created these ultra fast motors, and we said, well, can we learn and understand the rules, the rule book of how does one design a more efficient machine or faster machine, or a machine that rolling machine that maybe that doesn't roll as well or rolls differently. And so the two things came together. We said, look, we can build anything we want. What if we were to build random shapes where we can change the location of the DNA legs that are on the surface of the motor, on the surface of this chassis, even the chassis itself? We can change how flexible the chassis is by using the same design rules that DNA nanotechnology allowed. And so this opened up this idea that now that we can build things, we can build them and we can start thinking about their mechanical properties. Can we build things that are machines or motors from DNA building blocks? And so this led to this paper you mentioned, where we started creating many different shapes. And what we found is that if we create a nanoscale motor that is shaped like a roller pin, so essentially it's about 100 nanometers long. So human hair is about 100 micrometers long, so it's about 1,000 times smaller than a human hair. Its diameter is about 12 nanometers, So it's it's 10 times longer than it is wide, which is not different than the aspect ratio of a roller pin. And then what we did is we coated the surface of the roller pin with DNA, and we asked the question: what if one face of the roller pin lacks? We shave off the legs. Or what if we make a roller pin with a pivot in the center so it's floppy? What if it's a floppy roller pin? What if we change the density of legs on the roller pin? The DNA feet here. How does the speed change? How does the persistence, the processivity change? And from this, we came up with a set of rules that, by the way, led us to, creating machines that are now only a little bit slower than what the biological motors that nature evolved can do in terms of speed and processivity. So that was actually a really big step for us, and no pun intended, in terms of getting creating synthetic motors and machines that are getting closer to what nature is provided. Mm-hmm. And so with that, with those discoveries and with that work, that kind of led us to this general idea that the DNA mechanotechnology is akin to what the industrial maybe revolution provided in terms of creating machines that really transformed society today. And then thinking about how miniaturized machines have led to things like accelerometers and MEMS devices, various MEMS devices that are used in all sorts of applications. But the rules to create smaller machines and motors beyond the sub-micrometer length scale into the 10 nanometer length scale, those rules are not clear. And so the ability to build structures using DNA and to have them move, that is likely to become a critical step toward this idea of miniaturization and creating hopefully new technologies and new capabilities that were not available previously.
And this all comes to fruition in actually a couple of months ago, and this is when you publish in February Rolosen's mechanical detection of SARS-CoV-2 using DNA-based motor. This is the work that leads to Future Insight Prize, Merck KGaA, Darmstadt, Germany. And here, Khalid, it sort of all comes together in the sense that you, in this sort of proof of concept per Rolosense, you have this device, it's moving around, it's sensing, and it's not just sensing, it can sense pretty much any virus that you put in front of it on sort of how you train it. You also have the smartphone camera application, as you were mentioning before. Walk us through this, because obviously the folks at the Future Insight Prize got really excited about it. But talk a little bit about this work, because it's extremely timely, not just obviously for what we were dealing with the last couple of years, but for just broadly, as you say, enabling rapid, low-cost point-of-care testing for any virus.
Yeah, absolutely. So the basic idea was that, for all sensing, virus sensing or most sensing, is like thinking of chemists, how we sense things in analytical applications. It's often based on letting one thing bind another thing and then having some readout, electrochemical readout or a color readout. That's typically how it works. And for viruses, this is based on detecting either the nucleic acid, the RNA, for example, in SARS-CoV-2, mm-hmm. or you're detecting the proteins, and they're using something like a lateral flow assay or other assays like what you have in the home kit. And so essentially, that's how sensing is done in all disciplines. But interestingly, and this is again from my days as a PhD student, when I used the world's smallest pen. And what that small little sharp point can do is detect things, but based on mechanics, by feeling. Mm-hmm. So an atomic force microscope, much like you imagine your finger scanning along a surface, gets distorted. The cantilever bends up and down, depending on the mechanical forces that it experiences along a surface. And it's so ultra sensitive, you can detect single molecules using an AFM. Okay, and then this, again, not so different than how a record player, old school record player, will detect the grooves along the record, and as it distorts up and down, that creates music, of course. So we thought, this idea that mechanics is an untapped way to feel and detect biomolecules or detect anything, it led us to start thinking about how we can think about using mechanics to do detection or to do diagnosis, for example. The really, while we know how to do it using something like an atomic force microscope, using a single sharp point, we can't do it for many different sharp points at the same time. It's really a throughput problem. Is how do you do it in a convenient, facile manner, and how do you do it with sufficient throughput? And so when we looked at our motors, and in the years leading up to this discovery, we measured the amount of force that these motors generate, and we discovered that a single motor, one of these rolling DNA motors, generates approximately 100 piconewtons of force. Okay. So what is a piconewton, right? So we're talking. So for example, a paper clip probably has a force of gravity about a millinewton. Okay. So we're talking from 10 to the -3 to pico 10 to the -12. So a billion times smaller in force compared to the force of gravity experienced by a paper clip. A single cell will generate forces on the nanonewton. So this is 1,000 times smaller than the forces created by a cell. Okay, so 100, it's generating forces about 100 piconewtons. And the way we know this is that if we create barriers to the motor motion, and the barriers we created, we basically created a DNA duplex as a way to stall the motor. If we put a DNA duplex as a speed bump or a sticky patch for the motor, it can break interactions as strong as ones that require about 100 piconewtons. So we knew that if we created a sensing element that or a binding event that required more than 100 piconewtons of force, the motor would stop. And so we said, okay, how can we use this to develop a way to do detection of viruses? Well, the nice thing about viruses is they're, at least SARS-CoV-2 in this case, is it's studied with 20 to 30 copies of spike protein. And so what that meant is that if we created binding elements on the motor body and binding elements on the track that bound the spike proteins, we would get this Velcro-like multivalent binding to a single virus. And from many experiments, we confirmed that a single virus can stall one of these motors, binding of a single virus or bridging of a single virus between the motor and the track that stalls the motor. So the analogy would be like having throwing a wrench in a cog, or putting a stick in a rolling bicycle wheel. That's enough to stall the motor. And so, but the really interesting thing about it is that we don't need to break down the virus and pop open the virus and dig out the RNA. We don't need to break up the virus and isolate the antigens, the proteins on the surface. We are detecting the whole virion.
Okay.
And that actually represents a really untapped capability, because that's the most infectious agent, is the whole virion, and particularly the whole virion that's in the aerosolized form. And so this led us to a set of experiments where we started collecting exhaled breath condensate, okay, and showing that we can detect SARS-CoV-2 virions that are in exhaled breath condensate. Again, without any processing. We don't have an enzyme to amplify copies of the RNA or amplify copies of the protein. We are essentially using the virus as a mechanical bridge that shuts down the rolling motion of the machine. And the nice thing about this is that molecular interaction, the binding between the virus and the motor, leads to macroscopic or microscopic change, which is we see the motors no longer move. And that change in microscopic behavior can be detected using a simple camera reader, like one on a smartphone camera, for example, or even more simple ones. More recently, we've been building these readers using 3D printed microscopes, really tiny devices with a microprocessor that has built-in Bluetooth, Wi-Fi, and a camera and a CCD chip. So which makes the reader even more accessible compared to using a smartphone, in fact.
Mm-hmm.
And so this is one of the I would think in 2016 we had no idea that these rolling motors could have any applications in things like viral sensing, but this is what we've shown most recently, is that we can detect. And the really remarkable thing about it is, we add approximately 1,000 motors in a sample, and our limited detection is detecting about 1,000 virus copies. So on average, if we have one virus, one motor, that's enough for us to get a confirmation that that there's virus, that at least SARS-CoV-2 present in solution. The way we do it is you want to imagine, because our camera, using this simple low magnification camera imaging, we observe hundreds and hundreds of these motors simultaneously. We can watch them move along a chip. And it's kind of like a traffic camera watching cars in a busy highway. If you see the average speed of your car slow down, there's something causing that slowdown, and that's what leads us to a positive confirmation of the presence of the virus.
Mm-hmm. Yeah, it's completely fascinating, Khalid, and obviously, it was this project that got the interest of Merck KGaA, Darmstadt, Germany. You win the Future Insight Prize, and clearly it's obviously great to have one of the world's largest pharmaceutical companies interested in what you do, but sort of this early on, it's even more exciting because like a lot of these organizations that are transitioning beyond, hey, just pills or proteins or whatever, the future's in front of us, and there's new stuff coming. We've done a couple episodes in the past along sort of this theme of the electroceutical, and ultimately how, instead of using a pill, could we modulate a membrane potential on a cell instead of having the drug there. What is your future vision for, obviously we're in the early days, but nonetheless, you've come a long way the last couple of years in developing these tools. What is the the mechanoceutical, I don't know what you want to call it, I don't mean to coin my own word here, but what does that mechano intervention look like to you? Maybe look 10 years down the road for us, 15. What does that intervention potentially look like, where hey, we might not have this drug here, but we may have this little DNA device that's cleaning up the cell and fixing this and that and so forth. Take us into that future.
Yes, I've never been as delighted to receive any other recognition in my career. I think this Future Innovation Prize was absolutely a game changer for me. It really recognizes this whole field, and it really recognizes the quirky and unexpected nature of like basic discovery and how it could lead to different applications. You brought up this question of what can one imagine with the ability to control and generate forces? I mean, there's a whole range of future applications that one can imagine. In a paper last year, we showed that these motors can be used in computation. So essentially, we built logic gate operations. So you put DNA as an input, and then motion is the output. And so you can create logic gate operations, which are the basic components of how you create computers, AND gates, OR gates, and or gates, and you string them together, and now you've got the basic ingredients to get computation. The other really interesting thing is these motors can communicate together. So one motor can read an input, generate an output that gets detected by a neighbor. And when you have independent robotic elements, moving elements that can communicate together. That's, again, the basic component to creating new emergent properties, not so different than the behavior of things like social insects like bees and ants, which have very simple rules of how they can communicate together, input-output responses, and then you can get really complicated behaviors, really sophisticated behaviors from these simple rules where you have chemical input, mechanical output. In the area of diagnostics, I think the viral sensing is the most advanced phase. We envision a future where these types of devices can autonomously run, these motors autonomously run. We collect the aerosolized particles from the atmosphere, introduce them into solution, and essentially have this as just like you have smoke detectors inside every home. You can imagine viral sensors at every single busy train station or airport or hospital or school, passively monitoring the emergence of different viruses or different pathogens. The beauty is you get a real-time readout, GPS coordinates with time and space.
Yep.
And that could be a really important component to anticipating spikes in known viruses that are known to be circulating around the globe, and anticipate where future surges may be coming. But that's really, I think, the tip of the iceberg. I think this whole idea of mechanosensing, you brought up mechanopharmacology, detecting the mechanical state of a cell and then responding to that based on the release of certain drugs. We have some early versions of smart drugs that are triggered mechanically. You can also take it to a different angle. I mean, you have Michael Crichton's book Prey, for example, that predicted these nano-swarming robots that will come and haunt us. And I'm not saying that's what we have in store for us, but it's a really important step in a direction of starting to build synthetic systems that have some of the properties of living systems. And it's something I think we all dream of, having one day systems that can self-replicate and move and interact. And I think that would be an interesting direction in the future as well.
Yeah, I think that whole emergent theme per specifically tissue regeneration and sort of the 3D spatial, temporal-spatial development of new organs, and sort of where we have gone with this sort of beyond, in the sense of the sort of this, where the stem cell industry has gotten us so far, but you'll bring in sort of the physics of all this and create tissues and organs in the right temporal spatial configurations and all that. I'm excited for you. I mean, I think you have such an amazing future ahead of you in any direction you go with this. And it's just been, so exciting listening to what you've done so far. But yeah, the future is really bright for you. I clearly see why you won the award, and just really rooting you on, Khalid, as you continue to progress with this. Before I let you go, anything else hot for 2023? I mean, obviously been doing all the publications, but any upcoming talks or conferences or anything where we can listen to you, meet you, anything else on the schedule that you might want to mention? Please take the floor.
Well, as I understand, I'll be giving a lecture for this prize. It'll be live streamed on July 11th. And so that's one opportunity to listen in. I'll be focusing mostly on these DNA motors and their future applications. And that's, I think, a great venue.
Awesome. For everybody that is going to be listening to this particular episode of the podcast across the various networks or watching on the YouTube channel, again, you've been listening to Dr. Khalid Salaita, Professor of Chemistry, Emory University, Program Faculty, Department of Biomedical Engineering at both Emory and Georgia Tech, Program Members, Cancer Cell Biology, Winship Cancer Institute, and the recent winner, Future Insight Prize, Merck KGaA, Darmstadt, Germany, for its fascinating work in the domain of mechanobiology and DNA Mechano-Technologies. Khalid, I want to thank you again for taking the time out of your schedule to come talk to us, educate us on this fascinating domain. Obviously, thank you for everything you're doing for human health. And as we like to say here on our show, thanks for creating a better tomorrow via the work you do. Really, a really great story, and best for all of it as you move forward.
Thank you so much. I really appreciate it.